Well, hi, everybody, and welcome to the September edition of the Research Roundtable with Dr. Karen Herbst, who is our host and moderator. I'm Brenda Viola for Lympha Press, and I am delighted to do the introductions as always. It's such an honor to welcome Dr. Leon Straub. He is going to answer the question with Dr. Herbst, what are lipedema's molecular hallmarks? This is the central focus of our discussion tonight. And the research paper, which I have put the link in chat and will put it in the show notes as well, Defining Lipedema's Molecular Hallmarks by Multi-Omics Approach for Disease Prediction in Women, which was funded by the Lipedema Foundation, which we are so grateful for their support of emerging research and all that they do for the lipedema community. And of course, Dr. Karen Herbst did contribute to the paper as a corresponding author. Let me tell you a little bit about our guest, and I want to welcome all of you. You are logging on. Thank you for saying hello from wherever you are, and let us know where you are. We've got Utah and Missouri already. And as soon as I'm done the introductions, I'll put that link again for the paper we are discussing. So after undergraduate studies in biology, Dr. Straub graduated with a master's in science in life and medical sciences at the University of Bonn. Under the supervision of Christian Wolfram, he performed his doctorate studies and experimental work with a focus on modeling the influence of adipose tissue insulin resistance on diabetes development at the Laboratory of Translational Nutritional Biology of the ETH Zurich, which is a world-renowned and very prestigious place. And maybe we'll get a chance to talk about that a little bit. But after being the Walter Benjamin Fellow at the Touchstone Diabetes Center of the UT Southwestern Medical Center, he is currently a guest scientist at University Medical Center, Hamburg-Eppendorf. But we are so glad he is our special guest tonight. And as is the case, this is being recorded. Everybody who is registered will receive the recording. It will also be posted on lymphapress.com. We welcome your questions, everyone. Put them in the Q&A. I'm going to post that link again. And we've got Auburn, California, South Carolina, Aloha from Hawaii, San Diego, Raleigh, North Carolina, St. John's, Florida. Oh my goodness. And let's all buckle up and listen into a wonderful conversation. Take it away, Dr. Herbst.
Thank you so much, Brenda. And thank you for Lympha Press for sponsoring this research webinar tonight. And I know we're on a different Monday, but it looks like we've got a really good crowd tonight. So that's very exciting. And Leon, thank you so much for being our guest tonight.
Thank you for inviting me.
You're welcome. And I would like to focus on your latest paper. And can you, the title is actually quite long, but what it seems to be suggesting is that you're trying to make predictions for women who have lipedema, trying to figure stuff out, right? Using your multi-omics approach.
Yeah, thank you for pointing the attention towards our paper, actually. You're also corresponding author on that one. And I think it's the result of a very fruitful collaboration with you. And you have established this great cohort of patients in Arizona. And the clinical study has kind of allowed us to study in human tissue with blood and biopunches of the adipose tissue, the molecular underlying kind of pathways. And we will go into more detail, but so we used kind of a systems biology approach, which is to look from different angles into different molecular species. And then we try to find patterns that are true and correct across multiple angles.
So let's start with the beginning of the paper. One of the first things that was done was to look at leptin and adiponectin. And you found, I'm going to say you, got some interesting results for both of those. So could you first explain about leptin and adiponectin, what kind of hormones or adipokines they are, and then would you show us the data and show us the interesting finding that you found?
Oh, with pleasure. Thank you for that question. So, oops. Yeah. So this is the study, and the link is being posted again also in the chat. The adipocytes have been for a long while considered as a relatively inert organ, and also because of the work of Philip Scherer, who used to be my mentor in Dallas, and others. The hormones that the adipocytes produce and the lipid species that they emit into the circulation have been characterized, and it has been found that actually there is an important regulatory role for these cell types of for adipocytes to play in the whole body physiology. And here you see kind of different organs, schematically pointed out, and it is really widely across multiple organs. And we focused on first measuring these adipokines like adiponectin and leptin in our study, and because these are the predominant adipokines, and they have strong functions in leptin in kind of leptin and adiponectin in lipid metabolism and glucose metabolism. And so the question was, and that's why we measured it, was is maybe the adipocyte at the heart of that disease? And then we would expect the major signaling molecules to be changed. And now in this slide, I'm showing you the minus log 10 of the p-value on the vertical axis, and on the horizontal axis you see the fold change as the log 2 of the fold change. So that means how did the expression, and this is or not expression, this how much of this hormone is in the blood of lipedema patients compared to control subjects. And so if it's on the right-hand side of that black dotted line, then you see that it's higher in lipedema patients. And we adjusted the data of the measurement by the BMI of the different patients and the body mass index to be able to have a sensible comparison. And so we found that adiponectin is the higher molecular weight form, which is the physiologically more active form, as well as the adiponectin itself in a just one one protein are strongly increased in these patients, and leptin as well. So that is for us a key finding that we think it's actually the adipocyte at the heart of that disease in a way. And now on the right-hand side, you see the levels of these hormones and some other lipid species that become significant in the analysis, because so this kind of purple dotted line is where you can see what is significantly changed if you do statistical testing. And so we had also measured sphingolipids and ceramides, which are important lipid species that give information about not only adiponectin signaling, but also about potential roles they could play in inflammation and other processes that are connected to other tissues. And so if you, on the right-hand side, it's age adjusted. So if you adjust the data by age, then you see that also more of these lipid species come up. And so this was also an interesting finding.
Can I butt in here for a second? So adiponectin is an insulin sensitizing hormone. So would it be fair to interpret this data saying that because adiponectin levels are higher in women with lipedema compared to matched controls, that they have greater insulin sensitivity? Would that be
Yeah,
fair?
so that's what one could approximate when you know about adiponectin and the roles, but I think it's much more complicated. And adiponectin has been described to play this role in the liver and in certain other organs, but this question if lipedema women are more insulin sensitive and more glucose tolerant is a whole different story, which other people have studied to some extent, but I think there's no complete conclusive statement to be made according to what I know from the literature. There's also, of course, the whole question of what kind of fat tissue do you have as an individual? Subcutaneous fat could be more healthy than kind of the omental fat, but in general, this could be a hint for exactly that hypothesis as well that you put forward, and it could be that lipedema patients are on average more insulin sensitive, and adiponectin could be one of the factors mediating that. However, leptin has usually the opposite function. So, and we also see that increased. I think it's just also a sign that we're measuring that the adipocyte is kind of misregulating or is in this disease in lipedema. The adipocyte is at the heart of the misregulation of the physiological process.
Okay, so these hormones are a lot more complex than just a simple correlation with insulin sensitivity, and they also affect multiple organs, right? So
Correct.
that makes it complex right there.
Yeah.
There we go. Okay. Anything else about adiponectin and leptin you want to tell us?
Yeah, so the correlation I have here now, and so I think in general, what I would argue, also if other scientists are on the call, is that it's very important to see the whole complexity of the data, because what I showed you before were 2 dimensions, and now we have again 2 dimensions, but we have the individual points and we have added correlations to it. And I think what is important is not necessarily to just compare the absolute values, but also to see how they're distributing over the different individuals. And what we saw by doing that, and also resolving different stages as we have done here. So we have here stage 1 on the top left, and stage 2 lipedema on the top right, and 3 here, and then the control subjects. And if we would have now compared all lipedema patients with control, we wouldn't have found that, for example, that adiponectin is misregulated to some extent. And I think there is something to be said that maybe stage 1 of lipedema, if you say kind of it would be a chronic disease where most women start in stage 1, which of course is also a different whole debate, but then one could say that maybe the adipocytes and the misregulation of the adipokines and as a signaling molecule is important early on in the disease development.
I think that's really important. And I think staging is very important. And we actually just published a paper where we added some half stages in between 1 and 2 and 2 and 3, because sometimes there's such a big jump between the stages. And I was diagnosing women as, well, you're stage 1, but you're kind of 2. So I'd say you're a 1 to 2. And I did that so many times that we just converted it to 1.5, and we got a beautiful linear progression with BMI. So I think it is meaningful, and it may be helpful in research in the future. But you're right, those in stage 1, the higher the BMI, the higher the adiponectin, whereas in the other stages and controls, the higher the BMI, the lower the adiponectin. And that's super interesting. And maybe does that mean that as a woman with lipedema, maybe she gains some weight over time? Because it seems like lipedema drives the increase in, non-lipedema fat. And then that changes the whole picture. Whereas in stage 1, we're actually seeing true lipedema alone. Is that?
Yeah, exactly. And there could be other explanations. I think you hinted at kind of hyperplasia or the differentiation of new adipocytes. Then, of course, the location of the fat expansion can play a role in adiponectin levels also in the blood. But I would argue from what we present, I think it's really the misregulation of the adipocytes signaling towards the body. It could also, I mean, we correlate, we present here against the BMI. I think it's still one of the best measures. Of course, one could argue that other measures should also be taken into account, which is usually being done also in many studies. But it's hard to compare because the overall fat mass, if you have a high enough patient group, I think it's fair to use the BMI then.
Yeah, I
Because
think we're trying
yeah.
to add on waist hip ratio, waist height ratio, along with BMI, just to show that these women really don't have classic obesity, even though their BMI says that they do have obesity. So
I
that's
think especially in stage 1, the BMI is not such a poor measure because at stage 3 you might have a lot of more subcutaneous fat or fat located at different locations, and that could affect the adiponectin levels as well. But I think stage 1, that's why the finding that we have at stage 1, I find it very strong.
okay. So since we're on this topic, we have a good question from the audience from Valerie Mills, and she said, your data suggests a disruption of the BMI adiponectin relationship and early lipedema, raising the possibility of adiponectin resistance. Have you considered therapeutic strategies aimed at restoring adiponectin signaling, for example, targeting adiponectin receptor ceramidase activity or modulating ceramide metabolism? And could this represent a viable intervention for halting disease progression?
I think that's a great question. Actually, also other data of figure 5 that we have in that paper, and data on our ceramide levels that we measure in the blood of these patients actually are supportive of that hypothesis. And I think it's a great idea to follow that up, that research. Currently, I'm not planning any of that, but I think that's a really important research question. And because I will show that later, but we see actually most ceramides upregulated with lipedema. And we have both adiponectin, which is supposedly, or which can also downregulate ceramide levels in the blood. We have that up, but most ceramides are actually up as well. We have one ceramide, C26, I think, ceramide that is down, but that's the only species we see down. Yeah.
That's very interesting because it is I mean, we always talk about lipedema as a connective tissue disorder, but it's also a lipid disorder, right? Because adipocytes are full of fat, and I don't think this has ever really been looked at before. So I think it's a really great new avenue, and we need as many new avenues as we can to look at lipedema. So let's jump to the next topic. One of the things that you found was that when you're looking at the transcriptomic signature, that suggested that there was an upregulation in respiration, cellular respiration, and a downregulation of leukocyte pathways. Oh, hold on, I lost my.
Yeah. Yeah, great. Yeah, so I mean, that's was something that we didn't expect, right? So we thought maybe
Right.
we see some inflammatory markers up because often that is kind of measured, or you see fibrosis in some of the patients' adipose tissue samples, right, in the histology. Now, we did just messenger RNA, next generation sequencing, right, some transcriptome analysis, and here you see principal component plot of the results. So we had, I think it was 13 lipedema patients and 7 control patients. Sorry if it's not exactly the number, but roughly. And so so you see the from 2 different locations the data here. So we sequenced the RNA from both locations, abdomen side and from the side of the thigh. And so in red, you see lipedema samples, right? And then in kind of this blackish, in black, you see the control samples. And if you have to look at the dotted line, I think most of these go on their respective side if you say that they are distinct from each other, right? And
Yes.
so I think that was very promising when I saw that plot at first, and I thought, okay, we can find something now. The variability between the subject is very high. In human tissue, you often have that, but So we had to pool kind of for the analysis abdomen and thigh locations, but then we came actually to some interesting findings where we saw so you have like a list of genes that are differentially expressed, right? That's the outcome of this transcriptomic analysis of this RNA sequencing, and then you can analyze what kind of genes these are, and then you can also ask questions about which cell types do these genes belong to, right? And in the upregulated genes, we find many genes that are relatively selective for adipocytes, right, for adipose tissue. And then for the down-regulated genes on the right-hand side, you see that we see actually mostly genes for spleen, for kind of immune patterns, thymus, bone marrow. That's a niche for immune cells, right? Then blood, you have a lot of also leukocytes there. So that's already very interesting, I think.
Yep.
That means these cell types or these transcripts from these tissues and cell types are down. Then if we go one further, 1 second. Oh, am I stuck? Yeah, no, okay, sorry. And so now because you can read that in the paper, I just took the top 5 most gene ontology pathways. That means if you take all the differentially regulated genes, you can cluster them according to which pathways do they belong to that have a biological relevant function, right? So what could be their role in combination in changing the adipose tissue that we measured the RNA from? And on the left-hand side, you see the upregulated genes. Again, we pooled thigh and abdomen, and we found that aerobic respiration, cellular respiration, oxidative phosphorylation, and these things are the gene ontology pathways that upregulated genes belong to. And this means the mitochondria plays an important role in that adipose tissue. So you have more cellular respiration in lipedema fat than in control fat. And I think that's a very strong, actually, and I think that's very robust data. And then on the downregulated side, you have many genes that are downregulated in lipedema, in lipedema fat, and that have an association to leukocyte activation, endocytosis, and respiratory burst. This is all, most of the most highly associated pathways are actually in the immune activation. And I think that really tells us something about that it's actually not an inflammation that is increased in this lipedema fat. Now, you could say it's a punch biopsy, right? It's not necessarily in the nodules or in the tissue that is the most pathological in lipedema patients, but overall, when in the fat tissue or research, we expect that even surrounding fat tissue should have similar transcriptomic patterns. So it's usually described that when you have inflamed fat in some kind of locations in the depot, it spreads out and it goes also towards the edges. So I think it's we can conclude from that data, in my opinion. Yeah.
So what you're saying is that it sounds like that there's either a lot more mitochondria, or the mitochondria are a lot more active in lipedema tissue. And we know that mitochondria are increased in brown adipose tissue. Were you thinking brown adipose tissue at all, or is that not even relevant?
So during my PhD, I worked a lot on brown adipose tissue, and so I think it's hard to say. We also have in the CAC pathway, that's a different type of categorizing the genes for the pathways. We have thermogenesis actually that is upregulated, right? Thermogenesis pathway. And I think there could be a point to be made, and one should measure, in my opinion, lipedema patients. That would be a good hypothesis to measure their oxygen consumption rate, right, on baseline. One should study that, and that could have an impact on the metabolism and on other physiological processes of the patients, I think. Yeah.
But if they have more mitochondria, don't they have isn't the metabolic rate higher in the fat tissue then?
Yes, but I wouldn't say you cut out brown fat. I mean, most people don't have that much brown fat. Then you have something that's called beige fat. That's like brown adipocyte-like cells in the white
Mhm.
adipose tissue, and there could be some interspersed in the tissue that you took right from the patients. And but usually from the histology samples that I've seen, it seems more to me as if it's not
Right.
that much browning going on, but
I
I
agree.
could be wrong.
But could it be that the mitochondria that are increasing in number are dysfunctional in some way? So even though you're seeing more markers of mitochondria, they're just not working as effectively as they should?
Yeah, that's a great point. I think in general, when you're measuring things, often it's a compensation mechanism, right? But
Yes.
that could be exactly it. Um, One would have to measure the respiratory capacity of these tissues from the patients, right? I
Yep.
mean, one could also do that in like outside of the body, or the whole patient, but yeah.
And Valerie Mills is asking again if the abdomen's affected and the legs also affected, do you think that lipedema should be considered more of a systemic disease?
I think we have found some hallmarks that point towards systemic impact, potential one, and we have found kind of signaling molecules that could have this effect. We did not see any impact with regards to immune regulation on a systematic level, really. I mean, there's this trend for VEGF, vascular endothelial growth factor, to be
Mhm.
potentially upregulated. We saw a trend, maybe if you measure more patients, it would be significant. It was not. But also, previous studies have also found some VEGFs to be regulated in some patients compared to cellulite patients with some other adipose disease that is similar, as cellulitis, I think it's called. I don't know.
Mhm.
And and so the inflammatory markers, there's another study by Nankam et al. And from Pluyer's lab from 2022, where they measured some inflammatory markers in the blood that are up. I have a few doubts about that. I think they measured other important things, and it's a solid study, but I'm not so sure if inflammation is systematically up. We couldn't find that for sure in the cytokine chemokine panel that we measured of the patients.
So, we've there's a few papers that say it's an inflammatory disease, lipedema, and some that say it isn't. But this data goes along with it not being as inflammatory. And also, there's some papers saying that the macrophages that are seen in the tissue, which we're calling, inflammatory, are actually M2 macrophages, which are less inflammatory than M1 macrophages. And that seems to go along with this data a little bit better. Would you agree with that?
Yeah, I mean, the macrophages are just one of the immune cell types, right? So I wouldn't say it's all only about the question of M1, M2 macrophages, but yes, that is something that has been pretty consistently reported with the M2 macrophage type. So the M2 macrophage type is more of a tissue repair um, immune cell type, and it makes sense, but they also produce fibrosis in some way. But then sometimes it's really about the sequence of events, because they follow up on an inflammation, right? When tissue damage has been produced in the inflammation, usually they come later, M2 macrophages. I'm not so sure that one could just say, yeah, but it's just a tissue repair immune reaction. We also see some of the tissue repair hallmark in on the proteomic side, which we might come to. But with regards to the question of if it's a kind of inflammatory disease, I jumped over this a bit earlier, but I had this kind of in here, and I think, I mean, this I'm citing you here also because
Mhm.
so the classification of disease is EF 02.2, right? Also sometimes called lipoedema, and there are some important considerations regarding the classification. They are part of 14 in the international classification. It's the WHO classification. It's the disease of the skin, and then it goes down until EF02, where it says certain non-inflammatory of subcutaneous fat diseases, right? And then it's the lipedema. So I think in general, it has been categorized as a non-inflammatory disease of the subcutaneous fat. Now, one could argue maybe it needs to be changed, but from our data, we are in line with that. That's for sure.
It's so fascinating because, you know, I see women clinically and they come in and they say, God, I'm so inflamed. I feel so inflamed. And yet we're showing that they're not inflamed. And, you know, I don't think that at the time that we were doing these studies, we were, you know, questioning women deeply enough to find out whether they felt inflamed or not at the time. And I think that would be very interesting, is to actually listen to them uh, and they have flares, and at different times see how things change in their body. But let's
Yeah,
so
where does the pain come from also, right? I mean, that
yeah.
that could be also linked to the pain, the inflammation. Now, we don't find the evidence here. For me, with regards to, because as a scientist, I ask the question, what is the cause behind the disease usually? And we want to find not only biomarkers, but also kind of potential drug targets, right? And so I think many of the patients, you know it better than me, but it could be secondary effects, right? Either some adaptation, edemas forming, or even sometimes hematomes. And then so sometimes if you have early on during this disease development specific tissue process going on, it could mean that it does not prepare the tissue for disruption later on, and then you might have elevated inflammation in a situation where you have a hematome all of a sudden, or you have bruising going on, right? So that doesn't mean that this general function of the adipose tissue and of the adipocytes is anti-inflammatory. It can mean that having it at a kind of permanent anti-inflammatory state, or being prone for that role, or being wrongly prone, it's kind of maybe a disease cause, right? Could then lead to worse inflammation when it's the immune system and the tissue integrity is challenged later on. That's how I would want to see it. So also with regards to the question of fibrosis, right, or just the consistency of the tissue, I think you have a very heterogeneous tissue where you have nodules forming and you have all kinds of different processes going on at the same time. So I wouldn't say that. So for fibrosis, you usually need more inflammation also, or an immune cell activity. But I would say the hallmark is that there's less immune cell activity and it's suppressed in lipedema fat kind of causally. And then when it happens, like in the nodules, it could be like excessive, right?
Mm-hmm. It's fascinating. So this is your transcriptomic data, and this is a multi-omics paper. So can we move over to your proteomics data and tell me what you found there in terms of mitochondria and inflammation?
Yeah, so actually the samples that we had for the proteomics, they even clustered better. So it was more distinct. And here we have only, actually was Paul Longclass that who analyzed the tissue protein, and it's from the thigh. Excuse me. And so yeah, so this is a very promising clustering of the samples. That means there's something different. Now, so we have still large uh, variability. It's human samples, right? And they are not genetically identical, obviously, like we have it in mouse studies often. So, and then actually what we found, so these genes that are behind this gene ontology analysis that I'm showing you now, it was actually just very slightly over the significance line. So we were a bit lucky. And this is just to put it out for other researchers on human disease and also on lipedema. Sometimes you just need a higher N number. So try to include as many patients as possible, right? Always, and do analyze as much as possible. And yeah, so on the protein level, so in the proteomic analysis, it was actually similar what we found. And this is very much leading us to stronger conclusions then. But so mitochondrial organization and mitochondrial pathways were up, like were in the upregulated proteins. So they cluster in these GO pathways: mitochondrial organization, mitochondrial membrane organization, carboxylic acid metabolic process. That's also one that happens mostly in the mitochondria, and as well as confirming our transcriptomic data. From what I described earlier, we saw on the protein level also some mostly immune pathways um, in the downregulated proteins. So, like the most significant one is humoral immune response mediated by circulating immunoglobulins, and then complement activation. And this is still something puzzling, and maybe we want to talk about that as well. But that's something that I have not had connected to it yet, and that's something that is interesting in many regards, also because adiponectin sometimes seems to structurally resemble some kind of complement, like it shares some domains actually with the C1q, like some homology in some of its domains with that. And yeah, so in general, I think so we confirm our finding in the proteome. With the proteome, we've confirmed our finding in the transcriptome, and it's very interesting. Yeah.
I think that was very powerful. So just a quick question on the upregulated mitochondrial genes,
Mhm.
proteins. Were there any mitochondrial proteins that were not upregulated, that were down regulated, that you're not showing?
Yeah, so potentially, I honestly have to say I do not remember all the genes how they were regulated, but so the vast majority of the of the genes that are, you know, kind of associate or important for mitochondrial biogenesis, and these mitochondrial processes have been upregulated. So it's like these pathways are only called if there's a consistent upregulation of the genes that are associated with this biologically relevant process in the cells. So there's statistics behind it, and one can trust that the mitochondrial functions are increased most likely.
Okay. And then interesting thing about complement is that complement mediates the interaction between macrophages and adipocytes. So, and there's a lot of macrophages there in the tissue. So are they just there for cleanup, and they're not directly interacting with adipocytes? Kind of interesting.
Yes, but so we have less complement
Mm-hmm.
pathway proteins, right? And
Yep.
so these are really like complement factors, some of them, like C6 or some other C1R. And I mean, I went through them just recently to check, and it seems so it could be a consequence of immune cell prevalence in the tissue. But
Mm-hmm.
on the other hand, um, it could also be a local environment in which you have locally less complement. We have not measured it for the central blood, right, in the serum. It would be an interesting follow-up study to do to see if we find also less complement concentrations systemically, right? But there is this hypothesis that also Phil Scherer and also Ilya have put forward, right, in this Nature Reviews of Endocrinology, which is paper, which is that they say it could be connected, the cause for disease to pregnancy, because there you have hormonal events that could maybe lead to an activation of the complement cascade, right? Which I guess it's not so straightforward, and I don't want to contradict them, but I had discussed with him a little bit that I think then we would expect it to be upregulated. He is already happy enough that the complement activation is changed and these pathways are called. Now, it could be a compensation, right? That in the adipose tissue, less complement activity can be measured, while in other tissues or in other, like in the blood, it could be up. So I wouldn't want to conclude on that, but overall, I think it's fair to say that the immune activation is down in the fat tissue. That is something we can really conclude. And these different candidates and these different pathways on the immune side, and some of these factors that regulate the immune system, as you said, like we had already talked about adiponectin, now some complement factors are produced by the adipocytes themselves again. So I think
Absolutely.
studying the molecular function in tissue, in kind of modulation of the tissue is something that I actually want to do in the future, also with my own research and following up some of these candidates that we have there.
Valerie Mills is asking, do you think these transcriptomic or proteomic signatures are realistic candidates for a molecular diagnostic test? Or do you think
So,
something else is going to happen faster, like adipokines, ceramides, etc.?
so in our paper, we proposed a serum test because we were able, and I can show some slides more later if we come to that. But I think if you want to make a disease prediction, you need a higher N number to really model something well. And we were limited by, 6 and 6. So we would need to repeat that, or like add on to that study probably 4 times as many patients before we could model any predictive tools. And before saying something, it could, like some of these candidates and even the pathways, one could still say are interesting, yeah, candidates for any future test developments, but I would not be comfortable in saying that one measures just these pathways and then one can be certain about that it is lipedema, for example. So with regards to so we have some project that we actually want to put forward, some collaboration partners and in which we want to collect more RNA sequencing data and then have a very large number of parameters, but to model that, to do it statistically coherent and correct, you need also a very large amount of data of RNA sequencing from many patients. And so we are collecting data. There's already published studies out there that combined could get us close to the edge where we could then model something based on that. So I think on the transcriptomic side, especially because also the RNA sequencing done by private companies becomes more and more cheap, or it's the prices go down every year, and that's potentially some good diagnostic tool, I think, in the future.
So one of the other things that came out of this paper was the difference between women in menopause versus women who were not in menopause. And I think this is primarily in reference to lipids. Could you tell us kind of what serum lipid signature you found for women with lipedema in menopause versus not?
Yes. So I showed you similar figures before when we looked at adiponectin and adipokines. So here now I added on the top you have the ones I showed you before, and then on the bottom you have the same depiction with also the adjustment for BMI on the left and adjustment for age on the right, but with removing all the menopausal women from the analysis. So on top is all the patients and subjects that we had, and we compare here. We compare the change in the concentration in the blood, right? The fold change between lipedema patients and control subjects. And then on the bottom, we just remove from both groups the menopausal women. And so what you immediately see is that more kind of here you see that like lactosyl, sulfatides, hexosyl, ceramides come up, right? So it led us, actually, Karen and I, to then hypothesize or to kind of conclude and then put forward a new hypothesis that it could be that menopause covers up some of the serum lipid phenotype. And in all kinds of analysis, I would stress that it's important to distinguish between menopausal women or patients and pre-menopausal women. Um, so, so what causes lipedema is most likely that something that has something to do with the reaction or with the women's sex hormones. But Nankam and colleagues in their 2022 paper, which I mentioned earlier, with Plür, they also measured progesterone and estradiol, right? And I think that was solid data, and they found that there's no change. So between lipedema patients and control subjects. So I think I would argue it's the reaction to these sex hormones by the adipocytes that will create lipedema. Now, you have menopause is the time when in women the levels of progesterone and estradiol or estrogens drop down, right? So I think so they become like women lose their sex specific hormonal patterns. So they become, in some regard, a bit more like men. And we don't see many male lipedema patients, right? And so I think it's fair to say that we would expect this disease to already have developed before menopause, although some literature says also there are events during menopause that women report they have lipedema, but most reporting, actually self-reporting, comes also from puberty and pregnancy, right? So now I think it's a very important model to compare menopausal women to the other ones, because in the premenopausal lipedema patients, we still have this misregulated sex hormone driven kind of lipedema causing molecular effect. And afterwards, it should definitely not be the primary factor anymore. There could be still secondary processes on a biologically relevant level that we still measure, but they are definitely not the ones that should be kind of contributing to causing lipedema, in my opinion. So it's hard. I think I talked to Philip Krupa recently about study design, and it's an exclusion criteria, right? Often puberty, under 18. So often they are through puberty already. So they're not included in, the studies. And then pregnancy, you exclude pregnant women because of, yeah. So I think we need to go towards doing research with longitudinal cohorts. And I mean, it would be best to study in some of these maybe mother-child cohorts where we follow up the children for a longer period, and then I could even consider measuring our kind of lipedema serum panel in these children, and then see if we can trace lipedema development.
But I want to drive home the point that what you're saying is that you don't think it's the sex hormones themselves that are causing lipedema, it's the reaction of the adipocytes to the sex hormones. That's a big statement. And I want everyone to hear that.
But honestly, I mean, we have some evidence. I think it's not 100% conclusive, right? I mean, it's just
no. We've talked
it
about
needs
how
more studies.
the receptors could be different in the tissue, right? There's so many enzymes in the tissue that modulate pathways. So absolutely.
What do you think, actually, Karen?
I think you're right. However, I do think that in some circumstances, I have seen women who generate a lot of estrogen. And so in those cases, that estrogen could drive those pathways in the adipocytes further, and they may develop more advanced types of lipedema or different types of lipedema, not sure. But yeah, so I don't want to say that it's absolutely not, because there are the extremes, which we can learn from.
Also, then the adipocyte, if it reacts wrongly to lower levels of sex hormones, it could even react worse to higher levels, right? I mean,
absolutely. Yep. And, women cycle every month, right? We go from low to high estrogen over and over and over. So, um, maybe, what, and during the, like right before women have their period, they tend to feel very inflamed and they get cramps. And, so it may be that during different periods of the cycle, the different levels of sex hormones are affecting the adipocytes because of, specific changes, receptors, enzymes, pathways in the tissue, in the adipocyte tissue. So, because
Or
when
contributing to more variation in the measurements. Yeah.
more variation in the measurements, right? Because for this study, we didn't record like the day of their cycle. And that would have been, I think that's super important. I think measuring through the cycle of a woman would be very important, blood levels and doing
Mhm.
some transcriptomics through the cycle and comparing them to controls. And gosh, so much to do, right?
Yeah. Yeah, I would love to collaborate with you again, huh?
I would too. I would love to do some cycle stuff. So, we're 10 minutes away. Brenda's gonna appear any second. Is there anything else that you would like to tell us about your paper? One of the questions I had is like, why did you use AI for the paper? But if you have something you want to talk about, I'd love to
Yeah.
hear.
So this is the next slide. So I use machine learning to generate this predictive algorithm that we wanted to put forward to have a diagnostic way for lipedema. And yeah, so we used the serum, these lipid species in the serum. So we measured kind of some fat in the patient's blood, and then also adiponectin and leptin, and just a few adipokines. And
Mhm,
if you combine that, we were able to with machine learning, which is a form of using, and this is kind of sometimes called AI, right?
mhm,
Um, to have the computer be smarter than me, right? It's like, okay, so these models are actually smart computer kind of circuits where they have figured it out how these lipids in the blood need to be combined or regulated so that they would say with a high likelihood it is a lipedema patient. And we actually had astonishingly good results for some of them. So you see here three different model types. So there are different statistical processes that you can apply and different, with different structure model it. And so, for example, this random forest model that we had with a min-max scalar, it was so good that we only so here on the left-hand side, you see true label and on the bottom predicted label. So this model said so only in one case it thought that so in a control subject was a lipedema patient. So it's pretty accurate in some way. I mean, it could not predict that these five lipedema patients were really lipedema patients, but that's an error you are more willing to tolerate when you have these tests, right? And so I think that's pretty good and promising. Also, especially thinking about how we had it's good for the literature that's out there, but still a low number of patients, right? And
right.
yeah, so and also yeah, I guess it's promising with AI kind of tools, machine learning. We can try to develop new diagnostic ways of identifying lipedema patients, and that is very important. And this is just an overview of all the different species, and so it picked actually. Not, it didn't even, this model didn't even need all of these species to predict it. Like it just needed these here. And yeah,
Wow.
that's it. What I would, and thank you for your attention for everyone. And I would love to get more of your questions of the audience. And
There are a bunch of questions, so
stop sharing, or you want to know
you
on
can
the
you can
slides?
stop sharing. Yeah.
Okay.
Uh,
Multitude of questions, and a very engaged Valerie Mills. Thank you so much with those clinically in-depth questions. We can't get to them all. There was one that really kind of nudged at me, so I just want to bring that back up,
please,
which is,
please.
the integration of these findings with clinical care. How do we connect those dots? Because I think, we have both patients and clinicians in the audience. And what would be a takeaway, your main takeaway for patients who might be on tonight's webinar? Either of you.
Leon, do you have?
Okay, shall I go ahead? I mean, I'm
Please,
not an
yeah,
MD,
you're
but
the
I
star.
I would say you can be hopeful that soon there will be diagnostic tests based on molecular analysis, and I think that's very important because sometimes it's not easy psychologically also to be diagnosed, and then there are different opinions from different MDs also about certain diseases, and I think you can be hopeful, and we are working hard on finding the right proteins to target for you so that we will have drugs hopefully soon. And um, I think what one has to understand is it's very important to first really know what the disease is about
Yes.
before one invests all of this effort. And we are still laying the groundwork. It seems like, okay, why are you not already doing the drug testing, or why you're not developing faster? But I think sometimes you have to take a step back. And I think we did a milestone with that, Karen, and the other co-authors and I. And I think we're on a good way. And we'll do everything we can to support you and to get you healthy.
That's a very hopeful message, and I'm sure that you would acknowledge that as well, Dr. Herbst. Lympha Press puts these together every month to answer your questions and to really do a deep dive into the research. Honestly, to look at your paper, I needed you to talk through all of this
Yeah.
for me to really have a few aha moments. So I appreciate that. And I love when the research roundtable provokes interest in future studies. There was at least one occasion where Dr. Straub, you said, this should be something that we study in the future. How does that path happen? We want to shout out Lipedema Foundation for funding research. What was the genesis of this paper and how you got to bring it to publication?
Karen, you want to start? Or...
Um, we, the goal is to find something that we can use to improve the lives of women with lipedema. And the Lipedema Foundation put a bunch of us together, and we started collecting samples, and uh, different investigator groups had awards, and we passed out those samples. And the group that Leon was in with Dr. Scherer was actually doing things. And so we started, we gave them samples for the adiponectin leptin assays, and we were getting just strange results. And so we kept giving them more and more and more because we felt like if we gave them a lot more samples, we were going to better figure this out. And I think that came to fruition when we collected enough samples to get those stage 1 women, because they were hard to find. And then I really wanted to do proteomics, and other people were doing transcriptomics, and so we worked with a young man who really knows how to do mass spectrometry, and it took us
All on glass, yeah. Mm.
and it took us 2 tries to get it. The first try, the data was kind of wonky, so we collected more, and we had to get enough sample for him to process. And then, once we got the data, there's so much data, I didn't know what to do with it. And so it got handed over to Leon. So it, it's a, it was just a very big collaborative effort uh, amongst everybody. But the
Well,
goal
Yeah,
is
and I'm
to
very
find
thankful
Lympha.
that you. Oh, sorry. Yeah.
No, I was going to
Oh.
say the goal is to help these women.
Yeah, and I'm very thankful that you took me on board, and you took me, like, you were so generous in providing these samples and allowing me to be part of the project. Thank you very much, Karen, for that. And yeah, I think we do everything we can to support these women. I think it's very important to take their disease serious. And for me, I have to say, I learned so much about the clinical research side through that project. And I would love to work more on lipedema as a disease, and to find out avenues how to diagnose these patients earlier. And also then to find ways of how to molecularly target the diseased tissue and to and eventually heal it. I think this is one of my favorite project lines right now for my own academic career, and I would love to help heal these women one day.
Yay.
Yay. And I will
Yay.
just say in chat, Victoria Bowden said it so well. Thank you for your work and your dedication. It means so much to this community. And those are appropriate words to end with. As always, Dr. Leon Straub, thank you for honoring us with your presence. Dr. Herbst,
Mhm.
no, I don't think anybody could have asked those kinds of questions with your context and understanding. And this is why you are the anchor of the Research Roundtable. We look forward to all of you joining us next month. Dr. Nick Pennings will be with us. Join us on the first Monday in October. And with that, everybody, have a great night. Thanks so much.
Thank
Thank you,
you. Thank
Brenda,
you,
for the
Leon.
Thank
Thank you,
invitation.
you.
Thank
everybody.
you.
What a pleasure.
Bye.